Well, first of all, thank you, Barbara. Thank you for all of you people who organized this meeting. It's very impressive and very important for us at this moment. I think Brazilian science must appear for the audience and also discuss science in high levels with a lot of research. This is very important to keep our minds sane when the labs return their activity. So thank you very much for this opportunity, and I will talk a little bit about the behavioral response we measure in our lab, especially those related to alcohol abuse in zebrafish. And we have been working with serotonergic system for the last year, and also some responses involve oxidative stress related to mitochondrial function as well. So, to start this lecture, I will talk a little bit about alcohol abuse, which is certainly a public health problem worldwide. We have a high degree of mortality rates due to alcohol consumption, which is around 3 million of deaths per year, around 5.3% of the death from the entire world, more than tuberculosis, HIV, and AIDS, and diabetes. And alcohol abuse has lots of consequences related to alcohol-related disease, which include, for example, cognitive deficits. Alcohol can induce too much problems in memory, learning, formation, impulsive behavior, and also dependence when ethanol is consumed at long term. Due to these complex phenotypes involving alcohol abuse and exposure to ethanol in experimental models, they can be used to study a lot of phenotypes involving neuropsychiatric conditions, even in translational neuroscience research. When we compare, for example, the effects of alcohol with other drugs of abuse, we can see in this graph the overall harm score, including lots of problems related to drug consumption. For example, alcohol is the first one in this graph. We can see clearly a more impressive score when compared to other kind of drugs, for example, heroin and also cocaine and LSD. So it's very impressive the effects of ethanol can induce on biological systems. And the most part of the score are related to, for example, the economic cost for insurance and also for the government, as well as injuries related to alcohol consumption. The etiology of ethanol consumption is very complex and involve both individual factors as well as environmental characteristics. Among the individual factors, I can, for example, highlight genetics. Different genes are involved in alcohol-mediated response, brain physiology, and also the overall physiology of the organisms, and as well as brain functions. Between the environmental factors, we can highlight the peer influence and the social influence of people who drinks alcohol, the drug availability. If drug is available in some kind of environment, people can consume them more easily, and also stressful conditions, because we know Matt lecture yesterday show ethanol consumption has significantly increased in people who are these days with the lockdown condition. So this kind of features, both individually and also environmental conditions, are important to demonstrate the behavioral phenotype, are responsible for the behavioral phenotype of the individuals who drink alcohol. When we look at this graph here, we observe the biphasic effect of alcohol in humans, for example, which include both pleasure and also dysphoria. If people drinks few alcohol amount, the response is stimulant. People get more euphoric, for example, and had a lower risk of drinking. So if people stay drink for a while, for a long while, or even if the blood alcohol level reach more significant amounts in the blood, we can see clearly depressant-like effect, increasing the behavior we'll call dysphoria. So this response of ethanol include the increase of both GABAergic and glycine-mediated inhibition, and decrease of glutamate signaling, calcium influx, and the metabolic rate of organisms. And regarding the chronic responses of alcohol consumption, if people use a prolonged time consuming alcohol, we can observe probably a neuroadaptive response in which often develop tolerance. If people stop drinking alcohol for a while, we have observed and several data showing physical and emotional symptoms. For example, the withdrawal syndrome. Withdrawal syndrome, we observe anxiety, nausea, and also more damage effects, which include, for example, seizure conditions. When you look, for example, the response of both acute, chronic, and withdrawal response of alcohol, we see clearly the normal condition and adjustment between glutamatergic and GABAergic signaling. If alcohol is drank acutely, there is an increase of GABAergic mediated system, signaling, for example, and at chronic exposure, the NMDA receptors are overactivated, equilibrating or keeping the balance stable. If organisms are removed from or interrupted ethanol intake, we observe overexpression of glutamatergic system, showing the possible effects of withdrawal syndrome, described here above. And anyway, in various studies show other neurobehavioral response following ethanol consumption. For example, we have clear effects of reward systems such as serotonin and also dopamine. Both systems are involved in locomotion, cognition, aggression, and even anxiety behaviors. And the serotonergic pathway plays a key role, for example, in vicious cycle, impulsivity, and also motivation. Here we highlight some features involving changes in serotonergic signaling, which includes depression, aggressiveness, and alcohol abuse, which we will call this lecture. So we observe clearly the existence of a dose and time dependent effect in which lower doses increase neurotransmitter release, for example, serotonin and dopamine, which we call a positive reinforcement of this drug. And if alcohol is consumed chronically, we observe clearly a reduced amount of neurotransmitter release, which means negative reinforcement of these drugs. Even way, as Kyle Prevalent spoke with us, the serotonergic system in zebrafish is really different in terms of anatomical when we look for the brain of zebrafish, but is functionally conserved. What's this importance? Because we have serotonergic neurons in different brain areas. For example, here we see serotonergic neurons in zebrafish in pretectal population, or even hypothalamus, and also in nuclear raphe, for example. And positive markers of serotonergic neurons are observed in widespread zebrafish brain. And importantly, although the brain anatomy of zebrafish is very different when compared to the mammal's brain, we show high sensitivity of zebrafish to the most used serotonergic drugs, which are described even for rodent studies. Regarding the effects of ethanol on brain energy metabolism, we observed previously in our lab that acute ethanol consumption induces oxidative stress by increasing reactive oxygen species formation and also triggering lipid peroxidation, which is a classical effect involving membrane disruption due to the increase of this radical species here. So if we look for the mitochondrial, for example, ethanol metabolism increases a large amount of NADH, which is the reduced equivalent which go to mitochondria and pass their electron through the electron chain transport. and which culminates in reducing the oxygen to water. Anyway, if these electrons are not properly transferred to oxygen, we can induce more reactive oxygen species formation and even promoting more lipid peroxidation and cell death. And so it's possible that the ethanol metabolism, which increase in ADH formation, also promotes changes in mitochondrial from the brain. So I will not spoke a lot of zebrafish because everybody knows that zebrafish is a very suitable model organism to assess behavior, neurochemical, and also genetic features involving disclosure of a lot of drugs. But here in this timeline, we can clearly see that zebrafish has significantly increased its use after 1980s, possibly due to the morphological studies of larvae and also the genomic sequence of zebrafish already know. As we can see here in this picture, the genetic homology of zebrafish when compared to mammals in humans is around 70%. And these species show complex behavior, which allows the use of zebrafish models to assess, for example, learning, memory, and also other complex behavior. Importantly, zebrafish research has been increased when compared to rodents, basically because we have most protocols aiming to refine research, replace the use of high vertebrates in neurochemistry and other areas of knowledge, and also reducing the number of animal tests, which means the zebrafish research allows great obtention of data and very reliable data as well. As you can see in this picture here, I show you that anatomically different brain between humans and zebrafish, but the responses of alcohol is quite similar in both humans and zebrafish. For example, we observe that exposure to low concentrations of alcohol increases aggression and anxiolytic-like response in zebrafish as well, while high concentrations reduce, for example, locomotion, disrupt shoaling, and promote anxiogenic-like behavior, and also induces oxidative stress in the brain. And previous findings also demonstrated some neurochemical changes involving the monoamine and monoaminergic system in zebrafish. For example, dopamine and serotonin levels are often increased after acute alcohol consumption. We have shown that cholinesterase activity in the brain has also increased, possibly suggesting a role of the cholinergic system in alcohol-mediated response, and also a balance between enzymatic antioxidant defenses as well. Here, as Professor Linz Nadel previously shown for us, we observed the complexity of the zebrafish behavior. We can, for example, obtain three-dimensional reconstruction of the swimming traces using, for example, to assess the behavioral phenotypes of fish when exposed to different drugs. Here in this picture is a paper of our group I published during my PhD. I show you the swimming traces of a normal zebrafish in the novel tank test, which assess locomotion and vertical activity. You can see zebrafish swimming in the top and the bottom of the tank. And when we expose these animals to 1% of ethanol during 1 hour, 60 minutes, we show a significant decrease of vertical activity and increased immobility, showing possibly depressing effect after acute consumption of acute exposure of high ethanol concentrations. The results I'll show you today are referent to the work developed by our laboratory, especially those found by Talisi, which is my PhD student of the lab, and also one of the organizers of this meeting. This review has been recently published in Progress in Neuropsychopharmacology and Biological Psychiatry, was a collaborative review with Professor Parker and also Robert Gerlach, showing that zebrafish can be used, for example, for assessing different effects of drugs, for example, ethanol, also nicotine, and as well as other kind of drugs, for example, LSD, and as well. Here are the different tasks we can use for assessing zebrafish behavior. For locomotion and anxiety, we can use the novel tank test, the open field, and also like dark apparatus. For memory, we can use Y-maze, T-maze, the conditional plate preference as well, inhibitory avoidance task, social preference task, Scholling behavior to assess how zebrafish behave when we put in groups, and also to assess fear-like responses and aggressive behavior using the mirror-induced aggression task. These behavioral tasks we can use, for example, to assess the effects of ethanol in the brain, which, similar to humans, involve activation of inhibitory pathways and a decrease, for example, in excitatory pathways when ethanol is consumed acutely. Also, we have some other neuropeptides involved in ethanol-mediated response and neurotransmitter as well. So we can see clearly an evolutionarily conserved physiology in both humans and zebrafish, which can be assessed by different behavioral domains, which include aggression, locomotion, anxiety as well, which certainly increase the predictive phase and construct validities of our models. At the first moment, it's important to mention that low ethanol concentrations can acutely increase aggressive behavior. As Matt showed us yesterday, and also different kinds of behavior will show, especially those using the mirror, which zebrafish see their reflected image and tends to attack the mirror. This task reflects agonistic interactions with a virtual opponent, and zebrafish can approach, attack, and even display an aggressive profile. Here, of course, not a shark, it's the own image of zebrafish reflecting the mirror in which he tends to attack the virtual opponent. One limitation of this task, it does not reflect all natural interactions of diets, because you cannot see different kind of complex behavior we observe in nature, but it has a lot of practical advantages. For example, it's a non-invasive protocol in which fish are not placed to fight each other, they do not harm each other. It reduces the number of fish tests. Instead of 2 or more, we use a single fish per test. We have certainly a better control over the stimulus, because it does not involve, for example, chemical cues, but only visual, and tends to how fish, for example, see the opponent. And it's very impressive how pharmacological manipulations can alter or increase or decrease aggressive behavior, as we could see yesterday, the caffeine example. So in this test, we can measure the frequency and duration of aggression, which are relevant measures of this kind of behavior. So we have 2 rationales for performing this work and 2 main hypotheses. For example, the first one is we saw in the literature that ethanol can acutely increase serotonin content in whole brain preparations. So it's plausible that serotonergic system could play a role in this kind of behavior, including aggression and even anxiolytic-like response when acute ethanol exposure is performed. And importantly, both acute and chronic ethanol exposure promote oxidative stress in the brain. So if this protocols induces oxidative stress in the brain, central nervous system, it's plausible that ethanol also could affect mitochondrial function in our models. So our first question was, does the serotonergic system play a role in the acute effects of ethanol? Here's the paper, a recent paper we have published in the journal with collaborative data of Professor Adair from Florianopolis, which help us in this paper. The protocol was quite simple. First, we should replicate the previous data showing how acute exposure affects ethanol. Why we did this? Just to see which endpoints are influenced by acute alcohol exposure. So we put the fish individually for 1 hour in beakers with water, which means the control group, 0.25, 0.5, and 1% of ethanol in volume. And we tested the anxiety-like behaviors and locomotion using the novel tank test, and aggressive-like behavior using the mirror-induced aggression test. Our results were similar to those found previously, in which we observed that low concentrations of alcohol increase aggression, while high concentration do not affect this behavior. We did not see specifically changes in exploratory pattern in this task, only in terms of frequency and duration of aggression at lower doses of alcohol. When we put the animals in the novel tank, different data we obtained. For example, at higher concentrations of alcohol, we observed a reduced locomotion by measuring distance travel, turn angle, and increase of immobility, suggesting freezing behavior, and also significant decrease of vertical activity, showing less transition to top, decrease time spent top area, and also an increased latency to enter the top. So this data strongly reinforce that high concentrations of alcohol can really affect zebrafish behavior, which become more depressant. On the other hand, if we put zebrafish to lower alcohol concentration, 0.5, we can see increased time spent top area, suggesting anxiolytic-like responses. So these specific concentrations and these specific endpoints changed we use for measuring the influence of how serotonergic drugs could affect this behavior as well. Here is the main protocol used in our lab. We expose zebrafish to different serotonergic drugs, which include PCPA to deplete serotonin from synaptic cleft, and also to serotonergic antagonist, WAY100-135, which is a 1A antagonist, CGS 120-66A, which is 1B. 1B antagonist, and ketanserin, which is antagonist of 5-HT2A receptors, and also their respective agonists like buspirone, CGS 126B, and DOE. Experimental groups were exposed to these drugs and later exposed to ethanol 0.25, 0.5, and 1%. The agonist was used just to see if they could mimic the ethanol response we observed previously. So, as talked before, the novel tank test and the mirror-induced aggression test were used for the experiments. At first results, we could see clearly if we put zebrafish in the presence of alcohol here, we increase significantly the aggressive behaviors, as I told you previously, and the duration of aggression. But if we put treat zebrafish with PCPA, this response is abolished, both in terms of aggression and duration of aggressive episodes. So the main conclusion here is probably the serotonergic system plays a key role in aggressive behavior, because if we deplete or if we reduce serotonin in the brain, these responses are abolished. Anyway, we measured this response using antagonist and also agonist, and importantly, we see here that all antagonists clearly reduce ethanol-mediated response, but when we expose zebrafish to serotonergic agonists only, DOE, which is agonist of 2A receptors, mimic ethanol response. So probably, although the serotonergic system in zebrafish is quite complex and more receptors can exist, even in terms of genetic distribution and also in terms of paralog genes, we can clearly suggest that II-A receptors, this proof of concept data suggests that serotonergic II-A receptors play a role in aggression mediated of alcohol consumption. If we look the anxiolytic-like response, we can see that PCPA also reduces ethanol increase in time spent in top area. So probably serotonergic system also plays a role in anxiolytic-like effects of 0.5%. When we look the antagonist, we can see the receptor 1B. When we use the antagonist of 1B, this response of alcohol is clearly abolished. And when we put zebrafish in the presence of serotonergic agonists, we can see that CGS 126B, which is serotonergic 1B receptor agonist, no difference were observed when compared to ethanol-exposed fish. So ethanol response probably are mediated in terms of anxiolytic-like behaviors by serotonergic 1B receptors. Different than aggression, we saw the involvement of other kind of receptor, other subtype of receptor. And importantly, when we saw the depressant effects of alcohol using 1% exposure, all responses were quite similar to those found in ethanol group. So even if you use PCPA, the response were quite similar. So probably serotonergic system, we conclude that it does not play a key role in depressant effects observed when high ethanol concentrations are tested here. So these are the main results of the first part, showing a potential involvement of serotonergic receptors in both anxiolytic effects and also aggression. And our second point, our second question was, could ethanol affect mitochondrial bioenergetics in the brain? Why we perform this question? Because previous data, even from our lab, showed increased oxidative stress or changes in redox profile in zebrafish when exposed to different, especially 1% acute and 1% chronically when we put zebrafish for 8 days in the water after we measure some kind of oxidative stress related parameters such as lipid peroxidation and some enzymes as superoxide dismutase, catalase. We can see clearly changes, significant changes in terms of oxidative stress related endpoints. How can we perform this kind of experiment? We, as before, we put zebrafish to 1 hour for 1%, which induces changes in redox profile already shown in the literature, and subjected the samples, brain samples, to high resolution respirometry assays using the Ouroboros apparatus equipment. So we measured the oxygen flow in the samples by exposing zebrafish to different kinds of substrates, which affects mitochondrial transport electron chain. For example, if we put the baseline conditions, we can see zebrafish show, exposed to ethanol, show higher amounts of oxygen flow when compared to control unexposed fish. And if you look the oxidative phosphorylation mediated by complex 1, we can see a substantial increase of oxygen flow via probably via activation of complex 1. Here we uncouple the mitochondrial electron transport and also use rotenone and antimycin A to assess if other kind of systems such as complex 2 can also play a role in these effects, but no significant data were found here. Basically, we see this tree graph showing that ethanol changes some rerio. This rerio involves ROX-ETS, which is a kind of measure of ROS formation. Because we saw increase this measure, we presume that even mitochondrial function activated in acute ethanol consumption could increase ROS formation in zebrafish brain. the oxidative phosphorylation capacity increased in ethanol group, and also when we measure the succinate control factor, succinate is the substrate from complex 2, we saw a markedly reduction of these measures. So basically, ethanol increased ROS formation, as shown here. It enhanced oxidative phosphorylation, corroborating the higher bioenergetic efficiency, and also changes in complex 2 do not contribute to the effects of acute ethanol exposure. As shown here, basically the complex 1 is the main responsible for these effects. When we repeat exposure zebrafish to alcohol for 8 days, previous data show significant withdrawal effects after 8 days of exposure. We saw different results in terms of mitochondrial activity. For example, at the baseline, using endogenous substrates, we can see a reduction of oxygen flow, and also regarding the complex electron transference of complex 1 and complex 2, it was reduced as well, and also the complex 2 activity when we expose the samples to a rotenone, which inhibits the complex 1. So probably different than acute effects, we even seen changes in the rerio of complex 1 for complex 2, showing a major role of the complex 1 in this effects, and a facilitatory effect on ROS formation, because this rerio or ROX-ETS has also increased. So here are our hypotheses of this data to explain this data. Because ethanol metabolism in zebrafish and also in humans increased during oxidation NADH formation, this reduced equivalent could increase significantly when ethanol is metabolized, even in the Krebs cycle, when some precursor molecules are formed. And increasing this molecule here when acutely fish are exposed, it can activate complex 1 and then increase the mitochondrial production of ATP, probably corroborating this effect we observed in the increased mitochondrial bioenergetics when zebrafish were acutely exposed to 1% of alcohol. If we look closely for the chronic effects, the chronic effects were a little bit different. Ethanol, for example, inhibit the complex 2, and inhibiting the complex 2, it decreased mitochondrial function by inhibiting probably the ATP synthesis. And these changes in mitochondrial bioenergetics could reflect in increased ROS formation, because the electrons are not probably in a most efficient manner directed to the oxygen. So our main conclusions of these findings were serotonergic system probably mediates this behavioral response of ethanol following acute consumption, both in terms of acute exposure using II-A antagonists, which probably play a role in aggression, while 1B receptors facilitates, for example, anxiolytic-like behavior. And importantly, while acute exposure enhanced complex 1 mediated activity, we observed that chronic exposure to alcohol reduces complex 2 function. So different effects observed on complex 1 and complex 2 could also contribute to oxidative stress in the brain. And basically, overall, we highlight the use of these zebrafish models of alcohol administration, both acute and chronic, and to investigate the role of serotonergic and mitochondrial function as potential targets of ethanol, which are promising strategies for future studies related to both pharmacology and neurobehavioral research. So I'd like to thank my sponsors, the Federal University of Santa Maria here. If you don't meet our university, if you don't know our university, you are invited to come to visit us when the coronavirus syndrome condition pass away. And here are the team. We have lots of PhD students: Fabiano, Francini, Luis, Talisi. Talisi is the responsible for these experiments I show you: Vanessa, Flavia, master degree students, Julia, Chelly, and also several undergrad students. So this kind of work is possible because we have governmental agencies who give us funding. For example, CNPq, Conselho Nacional de Desenvolvimento Científico e Tecnológico, CAPES, and also from the state, FAPERG. So without them, probably these students will not receive funding and will not be able to exert properly the research in Brazil. Here is the most recent photo I have of the team. Sorry, Luis, by the picture with the mustache, but it's the only one I had, the most recent one, because we are in quarantine, so the lab did not stop. We keep doing science during this period, especially using the web conferences, using web applications to discuss articles and also to write papers. So Talise is this one who performed these studies I show you today. So I thank you everybody for the attention, and especially I hope everything could be fine. So please keep your mind sane and also keep yourself safe when this coronavirus pandemic state are around us. So everything will be fine, and hopefully as soon as possible we will reopen our activity in the labs. Thank you, Barbara.